Cell organelles. So what exactly is an organelle anyway? As the name suggests, cell organelles function in a very similar way to our own organs. They are different organelles with different functions, each coming together to create a healthy cell. Eukaryotic cells have a distinct nucleus and nuclear membrane, a major differentiating factor between eukaryotic and prokaryotic cells. Eukaryotic cells also happen to be larger and more complex most of the time. So what you'll see is with prokaryotic cells, they're generally smaller and more tube-shaped, whereas eukaryotic cells are much, much larger, and they tend to be in like circles and square shapes. Here's a diagram of a eukaryotic cell with a bunch of different organelles in it. I'm not going to read them all off, I'll just highlight a few. There's the plasma membrane, which you can see right here, that's protecting the cell. It's the boundary of the cell, and it's found in all eukaryotic cells. The ribosomes make where are they? There they are. Ribosomes make protein and help with protein synthesis. The mitochondria, as I'm sure all of you have heard, is the powerhouse of the cell. It helps with cellular respiration, and it has its own DNA. We're going to be talking a little bit more about mitochondria later. There's the cytoskeleton that sort of holds up the entire structure of the cell. There's the endoplasmic reticulum, that's sort of the highway of the cell. It's where a lot of proteins get transported. There's the Golgi apparatus, that's sort of like the post office of the cell. It can change and fine-tune certain molecules to make sure that they're good for the cell to use. And then there's lysosomes. Those are sort of like the garbage disposal of the cell. Alright, let's talk about peroxisomes. Peroxisomes are organelles that are membrane-bound and are responsible for the fatty acid breakdown in order to generate energy. They convert fatty acids and amino acids by oxidizing these substances, which gives the cell energy. The only bad part about the peroxisomes is that they often produce hydrogen peroxide, hence the name peroxisome. Here you can sort of see the structure of it. They have a lipid bilayer membrane and then a crystalline core that's not present in all the cells but is present in a lot of them. There's one minor problem. As I'm sure most of you know, hydrogen peroxide is not a good thing. It can be dangerous and even deadly for a cell as it will react quite easily with other compounds. But never fear, catalase is here. Peroxisomes organelles have about 50 enzymes, the most important of which is catalase. Catalase helps break down that dangerous hydrogen peroxide into something much safer and much more useful, which is water. There's some other functions of peroxisomes. Here I've quoted a couple of sources. Peroxisomes in the liver cells also handle the detoxification of many chemicals, including alcohol and drugs. Hopefully none of you are doing that. And then peroxisomes play two particularly important roles in plants. First, peroxisomes in seeds are responsible for the conversion of stored fatty acids to carbohydrates, which is critical to providing energy and raw materials for growth of the germinating plant. Second, peroxisomes in leaves are involved in photorespiration, which serves to metabolize a side product formed during photosynthesis. So another important thing to know about peroxisomes is that they're under the general category, which means they're in almost every cell pretty much permanently. Centrosomes are organelles that help build a cell's cytoskeleton. This is important because the cytoskeleton helps hold the cell together and gives it its structure. Centrosomes help build the cytoskeleton by providing the cell with microtubules, polymers that are an essential part of the cell structure. They really make up a lot of the cytoskeleton. Centrosomes are made of two smaller tubes called centrioles. We're going to look at those later, which come together to form a right angle. These tubes are made up of even smaller tubes in which 3 microtubules are stacked on top of each other. So you'll have like a stack of 3, so like you can put your fingers out and just put your top 3 fingers out. That's sort of what it looks like. There's a stack of 3 microtubules, and then there's 9 of those stacks in total forming this sort of other tube. So you can almost envision it as sort of like a Twizzler, but with 3 different microtubules in each stack. You can sort of see it here. This helps you visualize it better. Here's the stacks of microtubules. There's 9 sets of 3, which is 27 in all in the microtubule. You can see they're at the right angle. And then we're going to talk about this a little bit later. There's mother centrioles and daughter centrioles. That'll come into play when the cell begins to split and then replication happens. Centrosomes are temporary, and they're only needed to help build the cytoskeleton and to help the cells split chromatids from each other. They're about medium size and are in the cytoplasm of the cell. During the S phase of cell division, centrosomes can help change the structure of the cell to allow for a more smooth transformation. These centrosomes break apart and form new centrosomes, with each having one original centriole and one new one. So you can look here and see this mother centriole would be the original one from the centrosome, and then this daughter centriole is the newer one that was formed after the cell split. So you can sort of see that cycle visualized here on this graph. Mitochondria are some of the most familiar and important organelles in the entire cell, especially in heart muscle cells, which rely on mitochondria more than any other cell type. Mitochondria help provide the cell with energy and break down more advanced molecules into water and other more usable substances, similar to the peroxisome. During electron transport, the participating protein complexes push protons from the matrix, which is sort of the inner part of the mitochondria, we're going to learn about that a little bit later, into the intramembrane space. That's the space between the outer membrane of the mitochondria and the inner membrane of the mitochondria, as the name suggests. This creates a concentration gradient of protons that another protein complex called ATP synthase uses to power synthesis of the energy carrier molecule ATP. And as I'm sure we all know, ATP is vital to cell energy. A mitochondria shape, size, and a 21st century surprise. A mitochondrion is a medium-sized organelle with a pill-like shape. A mitochondrion has folded tubes called cristae inside of it, allowing for more surface area and thus easier energy production. Mitochondria also have two membranes, a semi-permeable outer one with pores, which lets larger compounds through, and a more restrictive inner membrane. The space between the inner and outer membrane, as I said earlier, is called the inner membrane space. These substances inside of the inner membrane is called the matrix. So the innermost part of the mitochondria is called the matrix. And similar to the nucleus, it's filled with DNA to help make proteins. New technology has allowed scientists to discover that instead of the cristae appearing as they do in most drawings and illustrations, that's sort of like the tube going down the center, it's really fat, they are actually attached to the membrane of a mitochondrion and aren't so centralized. It'll be easier to see in pictures here. You can sort of see this is what the general modern consensus of a mitochondrion shape is. As you can see, the tube is not as centralized and it's not as wavy as you see in the diagrams, and a lot of these cristae come up from the membrane versus something like this where it's just a tube that's winding up and down the middle. But these pictures sort of allow you to visualize the different parts a little bit better and help you sort of to see what's going on. Here's some fun facts about the organelles we've talked about today. Peroxisomes were one of the last organelles to be discovered. Centrosomes are only located in animal cells, they're not in plant cells, and then there are usually one or two mitochondria in sperm cells. And then another fun mitochondria fact that we're sort of studying is about the endosymbiosis theory, which states that about 1.5 billion years ago, in the fossil record, that's when we found these cells, scientists believe that the ancestor to our eukaryotic cells engulfed a mitochondria-like aerobic organelle, and the two lived in symbiosis. So you had this larger anaerobic cell take in the smaller aerobic cell, and the two were able to sort of help each other, and now we have eukaryotic cells. That's the theory that most scientists have today. Here's my sources. Thanks for watching.